Afternoon, everyone. I hope you can hear me and you can see my screen. Is everything fine? Hey,
Yes, thank you.
so it's great to see some of your faces online, although it's been a while since I've seen a lot of my friends that's on this meeting in person, and I hope it's going to happen soon. But I'm going to give a brief overview of what we're busy with in South Africa. And if you look at my slide, I play quite a few roles. I'm director of the Center for Viral Zoonosis, which is much broader than what I'm going to present today. But then I've also recently taken up a Future Africa chair, and I'll say something about that because that's strongly linked to One Health at the end of my talk. And then just maybe another comment, as some of you may know, I'm also the co-chair of OLAP, the One Health High-Level Expert Panel, and it may be good at some point for this network to share some of the thoughts from this committee going forward, especially in terms of One Health from a more global perspective, and also from the perspective where we're advising the WHO, FAO, OIE, and UNEP simultaneously going forward. See, my slides doesn't want to move. Okay, there we go. So the Center for Viral Zoonosis is specifically my research program. I'm a bat researcher. I'm trained as a virologist, but I do lots of other things. We have quite a large part on surveillance and developing diagnostic capacity, specifically for African applicability and low resource settings. But then the parts that I'm not going to talk a lot about today is the host pathogen interaction. So we still do experimental work and a lot of other work to really understand the host pathogen interactions. And I'm going to mention human-bat interactions, but we're really just moving into this field now. And then ecology of bats is really an integral part of our programs. So I'm going to specifically talk about one of the projects that we're currently busy with, and at a small part, just the coronavirus work on this project, which is a project funded by the American Defense Threat Reduction Agency. I'm the PI of the project together with Jonathan Epstein of EcoHealth. And it involves several role players in South Africa, the NICD, and then also Mozambique and Zimbabwe. So we're really looking at building capacity in Southern Africa, because we feel there's a lot of countries that's been neglected in terms of surveillance in the Southern African context. We always look at Central and Western Africa because of the history with filoviruses, but we've actually done very little in the Southern African countries. And then all our government departments are also part of this project. So it's really including everyone. It's focused on capacity building, lots of other viruses also. I will only talk about the coronaviruses, but it's really trying to implement One Health. So it's not just about surveillance in the bats, but it's really also starting to look at spillover hosts, which can be livestock, wildlife, and other humans. And then also bringing in human behavior aspects where we really want to look at the factors that may be important to understand risk of spillover, including environmental factors. So one of the advantages we have, because we live in South Africa and our labs is in South Africa, we really can do longitudinal surveillance quite often. We go to our sites monthly. We've selected sentinel sites based on risk. One is where there's a lot of religious activities and people go into these caves. The other one is a cave right in the middle of our Gauteng area in the city. And then we've got our fruit bat caves where there's agriculture and rural developments around these. So we really chose them based on risk. And we go into the field regularly, collecting all kinds of samples, but we also track animals by using things like the tooing. We've got pit tags, we've got radio tracking going at the moment to really also look at movement. And we really collect all aspects of the bat information that we can: age, sex, if they're pregnant, if they're scrotal. So we really combine our virology data with all of these things. And we've got a very strong connection with the National Museum. So bat identification and taxonomy is an integral part of what we do. We recently published a review on coronavirus surveillance to really just assess what's been done for Africa. And I'm not going to go into detail. We also made some recommendations about how future studies must look. But this is, for instance, one of the summaries with the darker colors showing the amount of studies that's been done. And you can clearly see that the SADC countries in Southern Africa, there's a lot of countries where we have absolutely no data. What we do know is the species of bats that's linked to potential zoonotic coronaviruses like 229E, NL63, MERS and SARS related do occur in these regions. So it's a bit important that we do look at surveillance. We've also looked at spillover studies, and this is extremely limited. The amounts look high, over 20,000, but if you really go into the detail, this has been very targeted studies, one species where there was actually disease or it was in a sanctuary, and that's the data that we have. So that's really a big limitation in most of the studies. And then if you start looking at human behavior, there's almost nothing that's linked to virology, ecology, the environment, and human behavior in one study. So our coronavirus biosurveillance look very similar from other countries. This is what you get. It's all over. So if you start testing bats for coronaviruses, you will get this high diversity. You see the patterns where certain viruses are associated with certain bat species. So they seem to be very species specific. And then you get the related viruses that's related to human coronaviruses like the sabecos, where we get SARS-related ones, and the mirbecos, the MERS-related ones. We've identified both viruses in those groups that's related to potential human viruses. Also SARS-related that's not yet published in rhinolophus. We also do longitudinal surveillance in fruit bats. We focus on the Rosetta aegyptiacus fruit bat, which is cave dwelling. And this is just, again, a very complicated situation that you will see. So if you start testing these bats monthly, you get viruses all over. And this is from population level fecal and rectal swabs from individual bats. And what was quite interesting, this is work that's currently in submission. But if you go and sequence the viruses, we see 3 dominant viruses in this bat colony. And if you look at the excretion patterns, they're not the same over seasons. So this just complicates the situation even more. They don't have the same peaks in the winter, depending on the virus that you look at. So just something to take note of. But what we see overall, and this is similar to some of the filovirus work done by CDC in Uganda and some of the other countries, that you see excretion patterns of coronaviruses that probably have something to do with juveniles and waning antibodies. And this is a very broad picture. We haven't checked this with any other methods. But then we also do see a peak in the winter months that can maybe have, it's got to do with some stress factors. So there's a low level of excretion throughout the year, but then we definitely have these high risk times that there's more excretion of viruses, specifically coronaviruses in fecal material. I'm just going to mention serology. We are working with USU on the Luminex system that a lot of you are familiar with, which excludes coronavirus surveillance, but we're actually busy making antigens for specifically some of the African coronaviruses. So we can do targeted serological surveillance, for instance, for HKU9 related viruses that we think is important, and then also some of the SARS related, but much more targeted, with the idea that we won't observe so much cross-reactivity. Some of the other things that I will just mention that I think is important for one out, we're looking at age. As a virologist, I'm involved in this, where we're really trying to find ways of better aging bats without having to do invasive methods like taking out tooth. So we're looking at scanned skulls and then also tooth wear, and then we try and see if we can correlate that with linear measurements in the field. And then in the future, we can only use linear measurements to do aging. We're tracking bats really to look at contact with human and other wildlife, and to also assess risk of spillover, diversity that they get into contact with. And then one of my recent postdocs are really looking at some of the environmental and land use factors that may constitute risk. This is a paper that's just been published where we look at land use changes around some of our very important bat caves in South Africa, and it's quite concerning. There's more than 10% people moving into these areas, increase in people moving into these areas close to caves in the last 10 years. And although we have legislation, it's not actually applied. So, and then human modification index, I'm going to stop now, Sharon. We also look at that and trying to overlay livestock with risk factors. We've developed several tools that I can share with you. We do education, and then Future Africa is a place where we bring all of this into a transdisciplinary space, bringing in other role players like policymakers, NGOs to really discuss all of this together. Right.